Okay, so in this lecture, lecture 11, we're going to get more into experimental design and cover a lot of aspects of controls and experiments and learn a lot of terminology. Before we get into the lecture, I just wanted to show you a couple of examples that kind of review what we talked about in our visualization lectures and in a study design lecture. We have some data here being presented. This is data that was shown before the Canada federal election in the year 2019, and it is showing job growth under two of our previous prime ministers, Justin Trudeau in his first term, and jobs created under Stephen Harper in his final term. And so it's a bar graph. It's showing us counts of thousands of jobs created. And it's a stacked bar graph showing you full-time jobs and part-time jobs. So there's several variables being presented here. And the question is, does this data provide evidence of causation? Here's yet another example. This is data that was presented in the campaign before the 2015 election. And this is a graph showing a bunch of previous prime ministers of Canada. The Trudeau on there is Justin Trudeau's father, Pierre Trudeau. Justin Trudeau hadn't been prime minister yet when this graph was made, and then Stephen Harper was the most recent prime minister there. And this graph is showing the y-axis is showing you a measure of how much of Canada's wealth was held by the richest 1% of the population. So a higher value on that graph indicates a greater proportion of wealth being held by the smaller number of people. Okay, and so again, does the data in this graph provide evidence of causation? And the last example along those same lines, this is data from the I don't know which year this data were published, but it came from the Ottawa Citizen, and it shows several different time periods with different prime ministers of Canada along the x-axis, and we're seeing a measure of average income for different segments of the population. Okay, and so what we're looking at in this graph is how income levels have changed over time for kind of different percentiles. And what this graph is showing is that the top 1%, that's the very top line on the graph, has really spiked in income in certain time periods, whereas people kind of in the top 10%, top 50%, and bottom 50%, their incomes have been pretty consistent over the past few decades. And so that's an indication of growing inequality overall. But the insinuation with this graph is that whatever political party was in power might be causing some of the changes in these lines. And I really want to question that, right? So this is an association, but it doesn't demonstrate causation. When we're looking at like data on countries and policies like this, we're not looking at controlled experiments. And in this last example, we can demonstrate that by looking at data from other countries. So this is a measure of income inequality. It's the share of total income going to the top 1%. And here we're seeing data from Canada as well as several other English-speaking countries. And you can see how the trends happening in Canada are also happening in other countries of the world. These are global trends. They're not the responsibility of like whatever one person is in charge of Canada at a particular time. And so this is an important point to make. Correlation or association is not enough to demonstrate causation. Okay, so we're going to talk today about experiments and how to design them. And I want you to remember that experiments are studies that manipulate a key independent variable of interest. And so suppose we have this research question, whether the colors of a peacock's brilliant eye spot feathers affect his mating success. This is something that I got to study as a graduate student. This is a peacock in my study population in the wilds of suburban Los Angeles. Okay, so let's test this with an experiment. We're going to manipulate the color of the peacock's eye spots and test whether that affects his mating success. For a treatment group, we'll add stickers onto every eye spot on males to alter his colors. And I really did this. I turned some of the males' eye spots a different color than they naturally are. And so an experimental group is a group that has some manipulation or treatment applied. And in my example, it was the peacocks with the stickers applied to their eye spots. In a medical study, it could be people who are given a newly developed medical treatment. The classical control group in an experiment is the group that just doesn't have that treatment. It's just the absence of that manipulation. So in my peacock study, I had peacocks in my sample that just didn't get stickers applied to them. And you might, in a medical study, have people who were just given no treatment at all. But what if the stickers weigh the feathers down? What if applying like 150 stickers to all these feathers makes it harder for the males to hold those feathers up and vibrate them the way they do for females? Well, this was a real possibility. So, in addition to my main treatment group and my classical control group, I had a negative control group. And that was a group where I applied the same stickers to the back of each and every one of the eye spot feathers. So it added the weight, but the color on the front of those eye spots was unchanged from its natural coloration. So the idea is that this negative control group accounts for the potential confounding variable introduced in my main experimental group. But I expect the males in this negative control group to have the same mating success as the males in my classical control group. Okay, so these are, this is getting at other types of experimental controls. So a negative control is a treatment that we expect to lack the effect of interest. A negative control allows us to control for confounds that may be important, and it's often the kind of everything but treatment. That's a way that I often remember it. The example of peacocks with the eye spot, the stickers on the backs of their feathers is one example. A really good example of a negative control is a placebo in a drug study. A placebo is this pill that's not expected to do anything, but it creates it produces all these other effects that taking a pill might have. A positive control is something that's more rarely seen in studies. A positive control is a treatment that's known to have some effect of interest, usually because it was shown to have that effect in previous studies. So you can think of the positive control as like a positive benchmark that we want to compare our treatment of interest against. Okay, so as an example, suppose you're testing a new drug that might treat some cancer, and our positive control would be people who are given an established drug that's already known to be beneficial for that type of cancer. That lets us ask whether this new drug is as good or maybe even better than the established treatment. These negative control, positive control distinctions can be tricky to remember. I've highlighted a blog post at the bottom here that I thought was pretty helpful. So let's look at an example and try to identify some controls in a real study. The example I want to look at has to do with plants that do really effective mimicry. And just to highlight the incredible diversity of what plants can do, we've got plants on this slide that imitate rocks on the bottom left to escape from herbivory. We've got plants that imitate female bees, including their scents to attract male bees and gain pollination. And then we've got plants on the bottom right that imitate dead meat in appearance and smell, and plants that imitate mushrooms. And that's what we're going to look at today, is the Dracula orchid. It's a flower that has a structure called the labellum that looks and smells like a mushroom. And it gets visited by flies that eat mushrooms. So could these flowers be mimicking mushrooms to attract pollinators? And so this particular research study that's linked down below was an experiment to test this question of what kinds of traits are important for attracting these flies. Okay, so they ran an experiment. In this experiment, they had a range of different treatments that included real flowers, and manipulated visual imagery of flowers, manipulated scents, and they even had 3D printed flowers. The ones I'm going to talk about today are these treatments shown on this slide. On the left in panel A, there was an experiment comparing the effect of real flowers attracting flies with real flowers in a bag. That's the visual only treatment. So the flies would be able to see it, but they wouldn't be able to smell the kind of mushroom-like scent. And then they've got a control condition there, which is just that bag on its own. On the B side of the slide, we've got another set of treatments. We've got the real flower again. We've got a bag that contained the scent of the real flower. So kind of the gases were collected from flowers and then presented in this bag. And then we've got a control condition, which was just the same kind of bag, but empty without any scents. And so these are some data. This is the figure summarizing the results of these experiments. On the y-axis, we've got the average number of flies per hour. And this graph is actually presenting two dependent variables. We've got the number of approaches and we've got the number of landings that the flies made on these different items. And it's shown as a stacked bar graph, which I think makes sense because I think to have landed, a fly must have also approached. So I think in this case, the stacked bar graph sort of makes sense. But let's just focus on the number of approaches, so kind of the overall height of these bars. Okay, so I just answered this one. What's the dependent variable here? It's the number of approaches that a fly makes to whatever it is, to a thing. And next, what are the independent variables? As a hint, there are two of them being presented in this particular figure. And then I want you to think about which treatment groups in this study and on this graph represent negative controls. And lastly, what can we conclude from the results being shown in this figure? What are the data telling us? Why was it important to test all of the different treatments in the same place or time per each real flower? So the way this experiment worked, on a given trunk of a tree, a real flower was presented alongside a bagged flower and an empty bag. Why was it important to do that in the same trunk of the tree at the same time? Well, it's because manipulations need to be balanced in space and time so that the control and treatment groups are truly comparable. If we did the study differently, let's say we measured all of the treatment groups on one tree, right? And then measured all of the controls on a different tree. Or let's say we measured all of the treatment groups on one particular day and then all the controls on a different day. Well, then location or time would become a confounding factor. And if fly activity varied in space or in time, our results might not be testing what we think they're testing. We'd be introducing bias by doing that. Okay, next example I want to look at involves a behavior of these macaque monkeys. So some macaque populations have a tradition of carefully washing their food. This short video shows some examples of macaques doing this. So we can ask a lot of questions about this. We might wonder, what benefits does it provide? Does cleaning their food help them avoid certain parasites? We might also wonder, what kinds of stimuli cause them to wash a piece of food? Do they wash every piece of food that they might eat, or is it only certain kinds of food? And what might prompt this behavior of food washing? And so that's exactly what researchers looked at in a recent experiment, looking at what kinds of factors would influence how the monkeys treated their food. And what they did was they put some tasty peanuts and things that the monkeys like to eat, and they put them on poop. And they put them on other substrates as well. And those were the different experimental groups. And so specifically, they had like a piece of food or a treat that was placed on an actual monkey poop, a piece of food that was placed on fake plastic poop, and then a piece of food that was placed on a plastic sheet. And so what is the dependent variable in this case? So what were they measuring in response to these treatments? What was the main independent variable of interest in this case? Is it possible that there's more than one here? And then what kinds of experimental groups do groups 1, 2, and 3 here represent, or treatments 1, 2, and 3 represent? And you can think of the main treatment group, classical control, negative control, positive control. Can you classify 1, 2, and 3 as these different types of experimental treatment groups? And lastly, what potential confound does group number 3 or treatment number 3 control for? That's food placed on just a plain plastic sheet. Okay, something to keep in mind. Not every experiment needs a control group. It all depends on what the research question is and what we expect to be important sources of variation affecting our dependent variable. Sometimes we're just interested in comparing two manipulations and we don't need a control group. Other times we really do need to compare a treatment group to a scenario where we do nothing or where we control for some kind of confound. Okay, so there's a whole bunch of terminology that you're going to need to learn to really understand experiments. And it's summarized on this slide. And I'll run through it really quickly here. It'll pop up again and again as we talk about experiments. Factors. A factor just refers to the independent variable in an experiment. Levels is used to refer to the specific treatments or groups within a factor. So let's say we're doing an experiment on vaccination. Our factor, our independent variable might be vaccination, and the levels might be people who got Pfizer vaccine or people who got Moderna vaccine. Okay, replication is the number of subjects or individuals sampled. How was the procedure repeated in the experiment? Balance or unbalance is something we'll get to later in this presentation. An experiment is balanced if the replicates are evenly divided over the treatment levels. If we have a difference in sample size in different treatment levels, then the experiment is unbalanced. Randomization refers to how subjects are randomly allocated to our experimental treatments. Ideally, any one subject should be equally likely to be allocated to any of the treatments. A blind experiment is an experiment done on humans where the information is hidden from the human subjects so they don't know which treatment group they're in. And a double-blind experiment is when this information is hidden from both the subjects and the experimenters who are conducting the experiment. Experimenter blinding can be used in other types of studies as well. So it doesn't have to be a human study. It could be, I could be studying the effect of hormones on monkeys, and I might have two injections, one that has my hormone treatment and one that is a control injection. And I may want to blind myself from knowing which monkey is getting which injection. And there's lots of ways to do that. And that helps reduce experimenter bias. Okay, so as some quick practice, what are the factors and levels in the Dracula flower experiment? And what are the factors and levels in the monkey poop experiment? And what would be the subjects or the replication in those experiments? Okay, let's look in more detail at how experiments can be designed. So here's a hypothetical study. Say we want to look at how fertilizer affects the growth of tomatoes. Okay, so just the title there alone should tell you what is the dependent variable and what is the independent variable. And say we have a couple of different options for how to do our study. We plan to do it in a greenhouse. Let's say we have 10 different greenhouses represented here by these 10 long, skinny rows. And each of the circles in this illustration is one of the plants. So we're going to run our experiment on 100 plants, and there's 10 tomato plants in each of our greenhouses. And let's say we have these two treatments. We've got our plants that we're going to apply a fertilizer to, and then we've got our classical control group in pink that is not going to get any fertilizer. So do you think this is a good design to design the study this way? We've got our two treatments, we've got 50 replicates, 50 plants in each treatment, 100 plants total, and we've got our 50 50 fertilizer plants in 5 of the greenhouses, and then our 50 control plants in 5 different greenhouses. Well, what about this option? Same parameters. So once again, we've got 100 replicates, 50 in each of our groups, but now I've got a different arrangement in the greenhouses. Now I've got each greenhouse has a mix of both treatments. Would this be better than option 1? And if so, why do you think so? What about this option? We could design our study like this. We could have each greenhouse have just one treatment, but we could alternate the types of treatments in this kind of blocked fashion. Or we've got option 4 and option 5, different ways of blocking our experiment. In option 4, again, each greenhouse has 5 and 5, but now we've alternated them. And in option 5, each greenhouse has 5 or 5, but we used a random number generator to assign where we were going to put these particular plants. And so we get this pattern where, again, still we've got 5 of each per greenhouse, but we've got random assignment. So we've got decisions to make. Blocking just means evenly distributing the experimental subjects or treatments according to some characteristic. And there's a saying about experiments: you should block what you can and you should randomize what you can't. All right, what is a balanced experiment? So we've mentioned this on the definition slide. Balanced means that you have perfectly equal numbers of experimental units or replicates or sample sizes in each treatment group. For example, if we had exactly 50 tomato plants in each of our 2 fertilizer treatments, that would be a perfectly balanced experiment. It's a good idea to aim for a balanced design. So think about why might that be. We can talk about that on Zoom. An unbalanced design just means we have unequal numbers of experimental units in each of our treatment groups. So suppose we had 80 replicates in our fertilizer group and 20 in our no fertilizer group. Would that be a good plan as compared to the 50-50 design? Why or why not? One thing to keep in mind is that very often experiments start out being balanced and then some units are lost unintentionally. For example, we might be running an experiment in the greenhouse and a few of our plants are knocked down and or just die. A few of the plants die and end up being dropped out of the study. It's very common to start perfectly balanced and end up with some slight imbalance just due to chance factors. Okay, what about experiments where we have more than one factor or hypothesis that we want to test in a single experiment? So suppose we want to test fertilizer, but we're also interested to know whether a particular insecticide might influence plant growth. What could we do? Well, we could run 2 separate experiments. We could have one experiment investigating the fertilizer, one experiment investigating the insecticide. Or we could run one experiment investigating both of these factors. So we could have a treatment for fertilizer, a treatment for insecticide, and we could have a control group that just lacked the insecticide and lacked the fertilizer. But we could also run a combined experiment that would investigate both fertilizer and insecticide and their combined effect. This is called a full factorial experiment. So this is an illustration of what that could look like. So in this case, we've got 4 treatment groups. In light pink, we've got no fertilizer, no insecticide. In light blue, we've got fertilizer, but no insecticide. In the more saturated pink, we've got no fertilizer, but yes, insecticide. And then in the saturated blue, we have both fertilizer and insecticide. And I've shown an example of how they might be distributed in a blocked fashion across our 10 greenhouses. This type of experiment is sometimes called fully crossed because we're testing all possible combinations of our different treatment groups. So what does the full factorial experiment tell you that the other two designs wouldn't? Well, think about these scenarios that I'm going to show you for possible results, possible data we could get from the experiment. Okay, so on the left, we've got one particular data scenario, and then on the right, we've got another scenario. So the colors are used to represent the different treatment groups similar to the previous slide. So what would the conclusion be in these two cases? And is this something that the full factorial experiment can tell you about that the other designs don't? Well, what we have going on here is called a statistical interaction. It refers to the scenario where the effect of one variable, insecticide in this case, depends on the level of variable 2, fertilizer. So if you dig into the graph on the right, what we're seeing on the right is that, when there's no insecticide, the plants with fertilizer grow a bit better. And when there is insecticide, there's a much bigger effect of the fertilizer, right? So it's like the effectiveness of the fertilizer depends on the insecticide. It's greatly enhanced when the insecticide is also present. By contrast, the graph on the left shows you that, the fertilizer is having a positive effect on the plant growth, but the insecticide is not having any effect. Whether or not there's any insecticide doesn't seem to matter. So this interaction effect is something that we wouldn't appreciate unless we had an experiment that had all possible combinations like this. Okay, so given the advantages of a factorial design, well, maybe all of our experiments should be factorial and we should just keep adding more and more factors. But this gets really tricky. Every time you add another factor into a factorial design, if you want to keep your sample sizes just as high, you end up having to double the number of replicates, right? Because what matters is your sample size per treatment group. So if we wanted to add yet another factor on here, we'd have to double the number of greenhouses, double the number of plants. Another drawback is that it becomes very challenging to interpret interactions between more than two factors. Interactions that involve 3 variables or 4 variables are just really difficult to understand. Okay, I want to talk about the difference between within subjects experiments and between subjects experiments. This applies to observational studies as well, but we very often see it in experiments. Okay, so sometimes experiments are designed so that all of the subjects experience all of the treatments in some order or other. This is called a within subjects design. When each individual can't be tested in every treatment, then the experiment has a between subjects design. That's when subjects get put into one group or another and they don't end up getting the treatment of the other group. So within subjects designs can be the most informative, but they're not always feasible. So for example, suppose we're testing the effect of a treatment that lasts a long time or that needs to occur during a specific window of time. Like imagine a study of reading in young children. And the question is whether kind of extra reading time might affect a child's intelligence later on. Well, we can't have a single child have reading time throughout its early life and then also have that same child experience not reading time, right? So we're studying something that's this long-term process. We can't have one child experience it both ways. Okay, so here's a few examples of within and between subjects designs. Sometimes within subjects designs are also called crossover designs because the subjects get crossed over into multiple treatment groups. So this was a study that came out a couple of years ago that was really interesting. It was an experiment on what people eat. And people were the adults in the study. There were 20 of them, and they lived in a hospital for several weeks to completely control their diet and to take a lot of measurements from them. And they were either given a diet of unprocessed food, so like fruits, vegetables, meat, things that weren't highly processed, or they were given a diet of ultra processed food, like, I don't know, pizza, hamburgers, things that come in packages that are super processed. And so every subject was put on a given diet for 2 weeks, and then they crossed over and experienced the other type of diet for 2 weeks. And at frequent moments, as illustrated in this figure, the researchers sampled things like their energy metabolism, their kind of components of that were circulating in their blood that are indicators of their metabolic health. And the 20 subjects were randomly assigned. So it was completely equally likely that any one person could be assigned to pathway number 1 or pathway number 2. And here's some data from the 2 experiments. So panel A, we're looking at a line graph showing time on the x-axis, so days on a particular diet, and the amount that a person ate in terms of calories. And what they found, so the people were they were given certain types of food, but they were allowed to eat as much as they wanted. And what they found was that the people on the ultra-processed diet, eating like pizza and cookies and things, they ate a lot more, presumably because these ultra-processed foods are processed to be super tasty and addictive. In panel B, there's some results measuring, again, the energy intake in terms of calories and kind of breaking it down into the types of macronutrients that people on these 2 different diets took in. And so that was a really interesting within subjects design. This is another example of within subjects design. This is a study that was done in Canada looking at whether the method of fluid intake for marathon runners would influence their performance and their physiology. And so they were comparing 2 different methods of intake. One was one was just drinking when you were thirsty, and the other one is called programmed fluid intake. And I don't know all the details of it, but it's something like you're kind of drinking more than you think you need and maybe more often. Okay, so in the paper on this study, they describe the subjects in their experiment, and in this particular table, they present some descriptive statistics with means, plus or minus standard deviation. I just want to highlight that as being a common way to communicate that information. And as a good practice, ask yourself, what kind of table is this, using the kinds of tables that we learned about last week. So these were the kind of characteristics of the people in their experiment. And the runners in their experiment had to run on a treadmill, and they both had to run under a thirst-driven treatment, and then they probably went home and came back on another day and ran in a programmed fluid intake treatment. And different runners would have done those 2 treatments in different orders. And what they found was that their running speed was not influenced by which type of fluid intake they were required to do. So it really didn't matter in terms of their running speed whether they were doing a thirst-driven or a programmed fluid intake. Okay, so here's a hypothetical example. Suppose we're studying the effect of a new drug called Soma on memory, and we want to compare it to a placebo, and we have 30 subjects that range in age from 32 years old to 59 years old, and we only have enough resources to perform a between subjects experiment. So each person is only going to be tested on one of the 2 treatment groups, either Soma, the drug, or the placebo. How should we assign the subjects to these treatment groups? So we looked at an example in the last lecture that was pretty similar to this. We could imagine 4 different ways of doing this. 2 of them not so good, 2 of them better. So a bad way to do it would be to just arbitrarily assign our subjects to different treatment groups. So this is like us attempting to be random. But as we saw earlier in this course, We humans are really bad at being truly random, and it's hard to avoid unconscious bias. We could imagine a badly designed study where the subjects were divided into groups by some characteristic like age, and then the two different age groups were assigned to the two different treatment groups. That would definitely be a bad way to do it. That would just introduce a major confound into our study. On the good column, we could use true random assignment to assign the subjects to different groups, and that requires using a computer and a random number generator. So we could take each subject and make each treatment group as a random sample of half the subjects. We could also use blocking, and that would be important when we are really concerned about a specific characteristic that might matter for our study. And we may wish to evenly distribute the subjects into different treatment groups based on that characteristic. So let me show you what I mean by that in a little illustration. So like imagine our sample is this set of subjects here, and these are their ages, the youngest one being 32 and the oldest being 59. There's only 8 here just to make this illustration a little bit quicker. Our confounding method might be, okay, let's put the young people in the drug group and the old people in the placebo group. Clearly, that's not a good idea. Blocking would be like this. We might take our subjects ordered by age and assign the youngest to the drug group, the next youngest to placebo group, the next youngest to drug, and then placebo, and so on. And if we follow that method, we'll end up with our two treatment groups being quite evenly matched in terms of their average ages. And so here's an example of the data that you get. So I kind of simulated this in R, and I created some graphs. So if we use blocking, here we can see that the distribution of ages for our two treatment groups is very similar. So that really removes age as a confound if we do this blocking. Now, question is, why is it often better to use blocking instead of truly random assignment? And as a hint, I want you to think about an experiment that has a particularly small sample size. So imagine an experiment with just 10 subjects, 5 of them are male, 5 of them are female, or 5 of them are old, 5 of them are young, and you need to assign them to these two different treatment groups. Would it be a good idea to use random assignment with such a small sample? And think back to our simulations where we practiced sampling. What goes on when your sample size is smaller? Okay, so here's a little illustration of what happens. Coming back to our drug and placebo group and using age as our variable here that we're worried about. So we could repeat random assignment, and it's going to be different every time we do it, right? So the first time I set up a true random assignment on my computer, the age distributions for the drug group and the placebo group look pretty good. But the second time I do it, just by chance, I happen to get a substantially younger group in the placebo group and a substantially older group on average in the drug group. And the third time I did it, the groups were pretty well matched in terms of their average age. But with random assignment, sometimes the groups might differ pretty substantially just by chance. And the smaller your sample size, the worse that problem is going to be. Okay, so the fourth time I did it, they were pretty evenly matched again. Okay, the last topic in this lecture is just to get into blinding. So blinding refers to a practice where study participants are prevented from knowing certain information that might influence them, thereby altering the results. So blinding is we usually talk about that for the subjects of a study. So usually it has to be humans, because like an animal won't understand necessarily. You can't explain to an animal the purpose of the study. But blinding can also be done on the researchers running the experiment, and it can even be done on people analyzing the data. And the reason why it's important to blind the experimenters and the people analyzing data is that we all have biases. We have experimenter bias, which refers to a process where we might unintentionally or intentionally influence the results based on our own preferred or expected outcome, or the thing we hope to see. And this process can affect all kinds of studies, observational studies, experimental studies, and others. So it's really important to think about ways to reduce the impact of our own personal biases. Here's an example of real data on why we need blinding. This is a table from a meta-analysis, and the study was looking at the relationship between, it was looking at studies in nutrition science and where their funding came from. And it looked at whether the funding source had any relationship to the type of conclusion that the given study made about a type of food. So an example would be like a study of Coca-Cola that might be funded by Coca-Cola and looking at the kind of health outcomes of drinking Coca-Cola. And so this meta-analysis collected information on a whole bunch of these studies, and they determined, was the funding source from the industry or was it not funded by industry? And they determined whether the study came to a favorable conclusion about that food product or unfavorable. And big surprise, every single study that was funded by industry, so looking at the left-hand column of this table, all of the industry-funded studies came to favorable conclusions about the food product they were studying. The studies that were not funded by industry had a closer to even split, where let's see 12 out of 19 of them came to favorable conclusions, 7 out of 19 came to unfavorable. So that's evidence that the funding sources that are supporting a study have a relationship with the conclusion of that study. And so that's just a more indication that we need kind of blinding. We need to seek out ways to reduce experimenter bias. As one final bit of practice, you should be able to say what type of table this is. All right, so just to wrap up with a reminder, homework number 6 is due on Thursday, October 21 at 4:30 p.m. And at this point, after you finish homework number 6, you are halfway through the course. And so that is really fantastic. And I hope you're all looking forward to reading week soon.